Do that. Do that. Good. Here's my audience, my father and my mother. We all know how easy it is to get rid of typos these days. Just a click or two and it's gone. Now imagine we could do the same thing for the human body. What if we had the ability to find typos in our DNA and get rid of them? The kind of mistakes that cause lifelong issues and just delete them. Now this may sound like science fiction, but it's actually reality with CRISPR technology. CRISPR-Cas9 is a revolutionary gene editing tool. This tool acts as a pair of molecular scissors, enabling us to modify our DNA. With CRISPR, we can treat a multitude of genetic disorders. I have spent the last couple of weeks analyzing and researching studies to find out exactly how this technology works and how it's reshaping the future of medicine. I'm going to explain where CRISPR came from, how it works on a microscopic level, and the serious ethical questions raised by its use. First, let's talk about the origin of CRISPR technology. CRISPR-Cas9 is an ancient immune system found in bacteria. These bacteria use this immune system to fight off viruses. When a virus invades, these bacteria will store snippets of the viral DNA and then create what are essentially wanted posters that they will then use to identify future viral threats and destroy them. This defense mechanism is what allows bacteria to survive even in environments rich with viral threats. According to a review in the National Library of Medicine, what began as a biological defense system for bacteria has now become the most effective, efficient, and accurate method of genome editing currently available. Scientists have realized that this ancient immune system can be reprogrammed to target almost any DNA sequence. These unique repeating DNA sequences were first discovered by scientists in the late 1980s. At first, they were originally thought to be pieces of junk DNA. It wouldn't be for another 2 decades that scientists finally realized that they were actually part of a sophisticated immune system. By 2012, researchers had finally harnessed the mechanism to create a programmable, easy-to-use gene editing tool. Now that we understand where CRISPR-Cas9 came from, let's talk about how we adapted this tool for human use. This system requires 2 key components. The first, the guide RNA. This acts as a GPS to find the target DNA. The second is the Cas9 protein itself, which acts as a pair of molecular scissors to cut the target DNA. The guide RNA is created to be a perfect match for the typo or specific DNA sequence you're intending to cut. Once the guide RNA locates and leads the Cas9 enzyme to the correct spot, the Cas9 snips the DNA. After the cut, the cell will try to repair that break, and during that repair process, scientists can either delete or add own genetic information. MIT Technology Review notes that the ease of design and high precision are why CRISPR-Cas9 has become the most widely used DNA editing tool in the world. While this technology has some life-changing implications, it also raises complex questions. A study in molecular biomedicine highlights the immense potential of CRISPR to treat debilitating genetic disorders such as sickle cell anemia. CRISPR can treat these disorders by correcting their underlying genetic mutations. While CRISPR technology has the potential to be one of humanity's greatest tools, it also raises some serious ethical questions. 2 researchers wrote that while CRISPR is beneficial, it forces us to address the questions regarding human intervention in our own biology. A major concern is the blurry distinction between therapeutic and enhancement applications. Another researcher argues that we must balance our drive for innovation with the need to protect future generations from lasting changes, such as any changes that will affect them harmfully. Now that we've covered the key aspects of CRISPR, let's summarize what we've learned. We have explored the bacterial roots of CRISPR, learned how Cas9 works at a microscopic level as a pair of programmable scissors, and finally discuss the ethical responsibilities that come with changing our genome. CRISPR has allowed humanity to go from passive observers of our genetic code to the editors of our future. As we stand at the beginning of a new scientific frontier, the question is no longer about how can we change our genetic code, but rather how will we do so responsibly. Thank you.